In this video, we will discuss the results of RMC-7977 in vivo, that is in the mice model. There are two kinds of mice model that they have used. First one is the immunocompromised mouse model and second one is the immunocompetent mouse model. And what they have found out is that RMC-7977 exhibit anti-tumor activities in the mice also. So let us understand what are the experiments that they did and how they come up to this conclusion. So they did experiments on human PDAC xenograft, and they use this human PDAC xenograft, and at the same time they also use this cell line derived xenograft and patient derived xenograft. There are two different kind of xenograft. First one is cell line derived xenograft, another one is patient derived xenograft, and they check the effect of RMC-7977 on that mice which are injected with this cell line derived xenograft or patient derived xenograft, and they use two different methods. First one is subcutaneous injection and another one is orthogonal injection, and these are the name of the source that is shown here. So, and what they have find out is that in all the 10 models they see the decrease in the they see the anti-tumor activity for all the 10 models. That can be seen in the panel A and also in the figure B, where we can see that when the RMC-7977 is injected, they see no flux of the tumor at the end point. However, the tumor regressions are shown in the 30% of the cases, and that is that information is presented in the paper, and I'm not sure how they come out to that 30% value from this figure. The next thing that they check to see whether RMC-7977 has an impact is that they do this for the orthogonal injection of the xenograft. They check the tumor volume, and they can see that in all the cases tumor volume goes down, and whereas in the vehicle the tumor volume goes up. And similarly they did check for the subcutaneous injection of the xenograft, and they see the similar results here also. And in order to check the side effect of this RMC-7977, they check the body weight change in the mice models, and they see that there is no statistically significant change occurs in the mice models, and it is more or less similar to the weight of the mice which are not infected or which are not injected with this RMC-7977. So this indicates that RMC-7977 has no impact on the body weight, whereas it has impact on the tumor volume and has the anti-tumor activity. So these experiments, that is panel A to panel E, are done on the immunocompromised mice models, and after getting the successful results or expected results, they did the experiment on the immunocompetent mice model also, and in that case also they see that there is a decrease in the tumor volume, but that decrease is not significant as they have seen in the immunocompromised mice models. So immunocompetent mice models also has the decrease in the tumor volume that can be seen here, and this is for both subcutaneous injection and orthogonal injection. And in the reason for this tumor decrease in the tumor volume change is that they find out that after 4 hours there is a decrease in the perk signal that can be seen here compared to the vehicle. There is a less perk signals in the RMC-7977 treated cells, and they use the dosage of amount as 10 mg per kg of the mice weight, and they see the decrease in the perk signal or phosphorylated ERK signals, and simultaneously they see the increase in the apoptosis, and the apoptosis indicator they have used is this CC3, the cleaved caspase 3, which is one of the which is the protein that is released or when there is apoptosis in which this is the protein which is produced when there is apoptotic apoptosis in the cells. So they can see that compared to the vehicle, the increase in the signal for the CC3 is high when the mice is treated with RMC-7977, and these results are after 4 hours uh, of this RMC-7977 dosage. And here also the dosage is same, that is 10 milligram per kg. So basically the figure 2 and figure 4 of the panel G are from the same mice models, and figure 1 and figure 3 of the panel G are from same mice models, that is the vehicle mice model. This is what seems like. So I hope you understood how they come to a point that RMC-7977 exhibits anti-tumor activity in vivo, as they did their experiment on the immunocompromised mice model and immunocompetent mice models, and both the models shows anti-tumor activity whenever this RMC-7977 is injected, either subcutaneously or orthogonally. I hope you understood the results that are presented in this figure. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.